Assalamu alaikum, my dear students. Today we are going to talk about mycelium organization or mycelium modifications. First of all, let's recapitulate what is mycelium. Mycelium simply means a network of hyphae. If you have a network of hyphae, they form a network-like structure. That structure is known as mycelium. Okay. Now, what is hyphae? Hyphae are slender thread-like structures or filaments of fungi. Okay. When you have a number of such filaments, they form network-like structure. That is mycelium. Okay. The mycelium in case of fungi, it is known to have different types of organizations or they are variously modified. The first modification that we are going to talk about is parenchyma. Parenchyma consists of loosely interwoven hyphae. Here you can see they are the loosely interwoven hyphae. The individual cells are individual hyphae. In this system, they are more or less parallel to each other. Here you can see this is one hypha, this is another hypha. They are more or less placed parallel to each other, and you can easily distinguish one hypha from another. Okay, they are easily distinguishable. So parenchyma consists of loosely interwoven hyphae, and the individual hypha are easily distinguishable from other such hyphae. The next one is pseudo parenchyma. It consists of closely intertwined hyphae, and these hyphae they form a parenchyma-like tissue. The individual hyphae in this case they lose their identity and are indistinguishable. Okay, so pseudo parenchyma में क्या होता है? The hyphae are rather closely intertwined. Okay, एक दूसरे के साथ बहुत associated होते हैं वो, and because of this fact they lose the identity. Now rhizomorph, it is root-like aggregation of somatic hyphae. The individual hyphae they lose their identity, and the functions of rhizomorph are they act as organized units and increase the infection ability of the fungi. Okay, here you can see this is the rhizomorph and this is the detailed structure of a single rhizomorph. Okay, the individual hyphae are so closely placed with each other that each hypha it loses its individuality and cannot be distinguished from other hyphae. Okay, next in series is Sclerotium. It is a compact and globose structure which is formed by aggregation of somatic hyphae just like rhizomorph. It acts as a resting stage because it stores enough food material and helps the fungi in vegetative propagation. This is the Sclerotium. It is a globose structure. The individual cells are compactly placed. They are closely aggregated. And this structure acts as a resting stage. It helps the fungi to override the unfavorable conditions because it stores enough food material inside it, and hence it acts as an organ of vegetative propagation in some fungi. Now the next one is Appresorium. It is terminal, simple or lobed swollen structure of germ tubes or infection hyphae. It is found in parasitic fungi like rustus and powdery mildews. Here you can see this is the spore. When it germinates, it produces the germ tube. The terminal points of these germ tubes they become swollen. Okay, they become swollen and sometimes they become lobed. These structures they are actually meant for the infection. Okay, they are actually meant for penetrating the host cells. Okay, and these structures, these Appresoria, they have been found in some parasitic fungi like rustus and powdery mildews. Then we have Haustoria. These are intracellular finger-like or branched absorbing structures of obligate parasitic fungi. The function of Haustoria is they absorb food from host. Here you can see these are Haustoria intracellular. These are the host cells. Okay, these are intracellular, finger-like or branched. Sometimes you can see the Haustoria are branched. Here they are simple. Okay, these are the absorbing structures. They help the fungi to absorb the food material from the host cells. Now the hyphal traps or snares. Hyphal traps are snares. They are actually sticky hyphae or loop of hyphae in some predaceous fungi. These structures, these hyphal traps, they help in trapping the nematodes. These are the hyphal traps. These are the sticky or loop-like structures which are made up of hyphae present in predaceous fungi, and they help these fungi to trap the nematodes. And then we have finally stromata. Stromata are compact somatic structures much like matrices. And these structures, these stromata, they form the fructifications on them. Here you can see these are compact somatic structures or mattress-like structures, and on these mattress-like structures the fructifications, fruiting bodies are formed. Okay, so these are some important mycelium organizations or mycelium modifications in fungi. This is all for today. See you soon with a new lesson on fungi. Till then, stay blessed and thank you.